thesis defense here. This is really a special sort of moment, both for her and myself, and I think Jacob Hooker, her co-mentor as well, with Emily being our first graduate student, and we hope not our last as well. So in that regard, Emily displays a very characteristic feature of those of you that know Emily, her braveness to go into areas uncharted and the unknown. Emily completed a Bachelor's of Science degree at the University of Arizona in 2009. She was involved in a range of activities, both on the side of chemistry and had a minor in psychology as well. She published work characterizing cell-penetrating peptides, work involved in bioactive lipids in moth biology, and perhaps the most interesting study she was involved in at that time, again reflective of her diverse interests, was on the crystallization patterns of ice on one of the moons of Jupiter. But one of the unique features that I thought captured my attention when reading Emily's CV when she approached myself about a rotation in the lab was her interest in understanding molecular interactions and characterizing structure and function of the nervous system. And I think that reflects her dual interests in chemistry, in psychology, neurobiology. And it's been a real pleasure to work with Emily over the past few years and watch her project evolve, her interests, and her continued passion and dedication to science. And I think Jacob could say the exact same thing. It's a pleasure to work with students like this who in the end come back and inspire us to do the work that we do. So with that as an introduction, the floor Emily's, and look forward to your talk.
Great, well thank you everyone for being here today. It means so much to me that I'll make it, especially to my family for traveling here. So today I'm going to be telling you about the bulk of my thesis project. I got the opportunity to work on many projects while in graduate school, especially being part of 2 labs, which is a unique and I think excellent opportunity to broadly sample what science can do. But so I'm going to tell you one particular story, and as you can kind of tell from the setup here, it spans a few orders of magnitude. I'm going to tell you about some small molecules that we discovered and characterized, and how they interact with a protein, both in a test tube in vitro, as well as in a more cellular context, and a little bit about the brain. And ultimately that's the goal here, is to learn about the brain and to develop, hopefully, or move towards development for development of small molecules to treat neurological disorders, and in particular, I'm interested in neuropsychiatric disorders. So although they are not as prevalent as disorders like heart disease or cancer, neuropsychiatric disorders are the most costly. And a large part of the reason why this is because of their relatively early age of onset. So the World Health Organization has this combined metric called the DALY score. And it measures the cumulative amount of disability caused by a disorder throughout one's lifetime. And what you can see from this orange curve is that mental and behavioral disorders affect the people that suffer from them very early in life. And because there are lacking effective treatments, this disability continues to accrue throughout the lifespan. And it's really only at the end that other disorders catch up in terms of the amount of disability that they cause. According to the National Institute for Mental Health, 50% of the people who will receive a diagnosis of a psychiatric disorder receive that diagnosis by age 4, very young. And then 75% by age 24. So what this tells us is that we need to be thinking about neuropsychiatric disorders as neurodevelopmental disorders, and we need to be thinking about them as the chronic diseases of young people. So what is happening in the brain during this critical developmental period? Here's just the generic developmental time window. This is the relevant time for the development of many neuropsychiatric diseases. But as one particular example, and we're going to focus in on schizophrenia, just to give you some examples. So any genetic risk that you have for developing the disease, you're going to get, obviously, right at the beginning. And then in addition, there are a large number of risk factors that appear to be relevant prenatally. These involve things like maternal malnutrition, stress, perhaps exposure to viruses, And this makes sense because there's an incredible amount of biology happening in development and in the brain during this period. New brain cells are born and proliferate. They grow in number dramatically. They migrate from the areas in which they're born to their final cortical locations. And they arborize. They send out neurite processes that they use to form connections with other neurons, and that will eventually lead to the circuitry that leads to cognition and other behaviors. In addition to this prenatal period, there's also a risk that is associated with early childhood development, particularly with adverse events. And in this window of time, one of the main things that's happening is neurons are being myelinated. The lipids that insulate the connections between neurons are being established. And then during early adolescence, the circuitry, especially in one particular part of the brain that's mentioned here, that's relevant to psychosis and to schizophrenia, the connections between the neurons get fine-tuned. And it's during this sort of critical stage that the first symptoms of the disease pathology that have been developing this whole time start to emerge. These aren't enough to necessarily diagnose today. These are things like maybe social withdrawal or disorganized thinking. But there are increasing risk factors in this range as well, particularly drugs abuse. And then sort of the culmination of this decade-long neurodevelopmental problem is really what we recognize as the first symptom. It's not the first symptom, it's actually very late in the disease. That would be something like a psychotic episode, the first time that a patient shows definitive sign of neuropsychiatric illness. And that's currently where we're starting our medical intervention. And then by the time you're only maybe 24 years old, you're now on a trajectory to have this disability for your entire life. So in order to shift our treatment paradigm, we really need to be able to intervene earlier in neuropsychiatric disease, ideally so that we can mitigate the amount of disability that's caused once the first critical event happens, and hopefully to be able to even prevent it would be the goal. We currently can't do this because we lack a comprehensive understanding of the genetic and environmental risk factors that lead to the disease and that originate in aberrant neurodevelopment. In order to be able to more finely pinpoint who is going to develop neuropsychiatric disease and who didn't, we still need a lot more science. On the basic side of things, there's been huge advances in the neuroscience field that are really driving a lot of, hopefully, improvements in clinical care. A lot of this is coming from the neuroimaging community, and it's allowing us to do things like detect who's at risk earlier and to stratify patients so that we can more accurately diagnose them. Whereas right now, we're mostly diagnosing based on loose clusters of clinical symptoms that appear to be common. And that will improve our clinical care. On the more pharmacology side of things, we really need better small molecule treatments. And not only that, but we need to understand how each person interacts with their treatment. And it may be that there are many different types of schizophrenias, and each one responds to a better response, have a different ideal treatment. So this idea of personalized medicine, how your exact genome interacts with the medicines that you take, can be critical moving forward in developing precise treatments. And then the part of the talk that I'm going to mostly focus on today is chemical biology. And that is a really cool field where we use small molecule tools to inform basic science, to learn fundamental things about the brain and the proteins in the brain, but also has translational power in order to develop new therapeutic options. And then just as a quick point, we're not going to totally science our way out of these problems, and there are a lot of areas for social reform that are obviously critical for tackling neuropsychiatric illness as well. So every time that in recent history that we've successfully developed a small molecule therapeutic to treat an illness, it's been based on a detailed molecular understanding of the underlying disease biology. And this is immediately challenging for neuropsychiatric illness because there are both genetic and environmental risk factors, and this, the biology that appears to go haywire is rooted in neurodevelopment. So how do you get a handle on this? Well, one area that we're very interested in and we think has the potential to generate new therapeutic options for these diseases is epigenetics. And that's because epigenetics regulates the expression of genes, so it interacts with your genetic background. It's sensitive to environmental changes, so exposure to toxins and stress can impact your epigenome. And it's critical for development. So it sort of fits the bill, so to speak. So the canonical way to approach epigenetics is something like this, where you have a couple of different pictures of cells, and then what you'll realize, and you'll also realize this looking at your own body, is that all of these cells have the same DNA sequence. They're all from the exact same person, but clearly they have different morphologies and different biochemical properties, different proteins are expressed. So although they have the same genome, they're able to do this because they have different epigenomes, different patterns of genes that are expressed, resulting in different cellular phenotypes. That's really cool. But what's even cooler now is if I tell you that this first image is actually from a patient that had a skin punch in their upper arm, or shown here, and that these are the fibroblasts that resulted from that skin punch. And then by application of a select number of molecules, you can reprogram the epigenome of that cell and turn it into a pluripotent stem cell, as shown here. So by changing the epigenetic landscape of a cell without changing its genetic landscape, you can change its cell type. Not only reprogramming it to a pluripotent cell, but then redifferentiating it first through a progenitor cell, which is on its way to becoming a neuron, but not quite committed to a neuronal lineage. And then finally, all the way to functioning neuron. So this is remarkable technology, and this is some of the technology that we're using in Integrity Lab and many labs are using to get at that personalized medicine piece of the puzzle, where we want the exact genetic background of a patient, but we want to study their neurons. So one of the things that epigenetics is so important for is maintenance of cellular identity. So if you are a pluripotent stem cell and you're dividing, you don't want to have a daughter cell that has the incorrect epigenome and suddenly turns into a different cell type. So for mitotic cells, this is critical. You need to make sure that the daughter cells end up inheriting the correct epigenome from the parent. And then really specialized cells, neurons, have actually exited the cell cycle. They still have all this epigenetic machinery, but they don't have to worry about passing it on to a daughter cell. Instead, they can really fine-tune their epigenetic response to the environment. And this is something that we know neurons do exquisitely. And they have to, because neurons are exposed to a tremendous array of different inputs from the environment that they have to deal with. These involve things like signaling molecules, like proteins and secreted factors from other neurons and also in the body, as well as neurotransmitters, the small molecules that mediate interactions between neurons. And then more generally, environmental exposure to things like toxins, stress, oxidative stress, and metabolites all impact and are sensed by neurons and transduced to the epigenetic machinery via signaling pathways in the cell. Then the epigenetic machinery, upon sensing, broadly speaking, these stimuli, modulates gene expression, and then that affects the downstream response in a very profound and general way. This will include things like how new connections between neurons are formed, or how they're strengthened in the case of memories or forgotten sometimes. It regulates things like addiction and mood, and how new neurons are generated and integrated into existing circuitry in the brain in the process of neurogenesis. So we know that these processes, as schematized here, are important for many, many aspects of neurobiology. And there's two main lines of evidence for this. First is that there are known mutations, both in the signaling pathway, proteins in the signaling pathway, and not just proteins, I should say, as well as in the machinery that regulates the epigenome itself. And these are associated often with profound deficits in these sort of downstream factors. In addition, we know that we can target the proteins in signaling pathways and that directly regulate the epigenome with small molecules, and that these too have impacts on broad neurological phenotypes. So I've gone on at some length now about epigenetic regulation without really giving you a molecular picture. So let's take a look at how it looks at what this epigenetic regulation actually kind of looks like. Still very schematized here, but getting more detail. So within the nucleus, DNA is compacted around chromatin fibers and wound around histone proteins in this core unit called the nucleosome that's used for packaging, as well as to regulate the accessibility of DNA to things like transcription factors and other regulators. Both histones, particularly their N-terminal tails, and DNA can be post-translationally modified. And histone tails in particular are extensively post-translationally modified. And these marks are deposited, they're read, and they're removed by multi-subunit protein complexes. And together those coordinate to regulate the expression of the genes that are encoded in the DNA underlying the nucleosome structure. So here's one cartoon schematic of the nucleosome. Here's a slightly more refined cartoon schematic. This is from the crystal structure of the nucleosome. Again, you can see DNA wrapped around. Now you can kind of more clearly tell that these are histone proteins. It's octamer of 4 to 5 sets of dimers. And one of these in particular that I'm going to be telling you more about is histone H3. And you can't see its tail here because the tails are not crystallized because they're very flexible. But here's its N-terminal tail sequence. And many of these residues are post-translation modified. There's a variety of different marks. The one that I'm going to tell you about in particular is histone lysine methylation. So within this histone tail, there are multiple lysines. So for example, if this represents the lysine at position 4, this would be H3, K4, and then the methylation status. Here's just a normal lysine residue with a side chain. And you can see that it can have mono, di, or trimethylation. And each of these marks, depending on the extent of the methylation, where it is on the histone tail, and what the other marks around it are, will have different effects on the gene expression. So this is a very fine-tuned process with potential for a lot of precision. So these methyl groups are deposited and removed by enzymes. The enzymes that deposit methyl groups are called methyltransferases, and then they're opposed by enzymes that remove methyl groups called demethylases. And we can exert some amount of chemical control over this process by coming in with small molecules that target these enzymes. So for example, a small molecule that might inhibit a demethylase. Okay, so here's the sort of molecular detail. So let's now take a step back and reconsider the question of neuropsychiatric illness. So we know that there are genetic and environmental risk factors. And one way that scientists have attempted to figure out what the relevant risk factors are is by taking a large number of patients with psychiatric disease and looking across their genomes and saying what variants, naturally occurring variants that are found in the population associate with the likelihood of having disease. It's not that they cause disease per se, it's just that there's a natural variant in that gene, and if you have that variant, you're more likely to get a neuropsychiatric illness. This is a very recent analysis. It actually goes one step further than that genome-wide association study. Now it's saying, okay, forget what the actual target is that appears to be associated. What does that target do? And if we look at a bunch of different targets that appear to be associated with neuropsychiatric diseases, do they have any functions in common? So this is called a pathway analysis. And it gets away from looking at the specific protein that has a variant itself and looking more at the overall process, the biological process that's being affected. And when you do this, what you see is that histone methylation appears to be the most statistically associated natural variant that co-occurs with psychiatric disease broadly. This is an aggregate score for 3 major psychiatric diseases. Not just histone methylation, but in particular histone H3K4 methylation seems to be the most highly associated, particularly with bipolar and with schizophrenia. Okay, so I just want to take a second to recap sort of the big intro. We really need new treatments for neuropsychiatric disorders, and we think that epigenetic mechanisms might be a good place to look. That's because epigenetic mechanisms involve the genome, they involve the environment, which I'll tell you a little bit more about later. And statistically, they're associated with disease. Furthermore, epigenetic regulation is affected by enzymes. These are targetable. So this is something we could actually come in with a small molecule and change. But there are a lot of unknowns, and so that's why it makes sense. So the rest of my talk is going to focus on one particular enzyme, and it's called lysine specific